In Lecture 5, we will learn about how twin studies can be used to calculate heritability. But first, back to our fundamental law of genetics. Phenotype equals genotype plus environment. In the last lecture, we learned about the formal mathematical representation of this law. Phenotypic variance equals genotypic variance, which is the contribution of genotypic differences to the phenotypic variance, plus environmental variance, which is the contribution of environmental differences to phenotypic variance. And remember the key assumption that genotypic and environmental variances are independent. Before we start, let's finish question 4 on our term papers. We already got a start on this question in last lecture. What genetic factors contribute to your complex trait? Write down 2 or 3 genes that you expect to influence your trait. You can look up the genes on the internet and make sure you cite your sources, or you can speculate or hypothesize about genes you think would influence your trait. The second part is what environmental factors contribute to the trait. You answered this question last Tuesday and now can work on polishing up that answer for 2 or 3 environmental factors. Finally, explain how the phenotype of your complex trait depends on the genotype and environment. Okay, back to our fundamental law. Now, phenotypic variance, this term is just measured. Just like we can measure the heights of a population of people and calculate the variance of those heights. How are we going to estimate the genotypic variance and the environmental variance? As was mentioned in last lecture, identical twins are expected to have no genotypic variance. Their genotypic variance is 0. And so any phenotypic variance is due to environmental variance. And so by looking at identical twins, we can get at this term. Then one might expect by comparing the phenotypic variance of identical twins to fraternal twins, we might get insight into the genotypic variance term. The environmental variance of identical versus fraternal twins is expected to be about the same, whereas fraternal twins have a positive genotypic variance term, which as we said is 0 for identical twins. By comparing these 2 sets of twins, we can get insight into the genotypic variance term. Here are some quick facts about twins. They represent about 1% of all births, split roughly evenly between identical twins and fraternal twins. Identical twins are also called monozygotic twins, which is abbreviated MZ. These twins arise from a single fertilized egg or zygote, which is why they have identical genotypes. This egg splits very early in development into 2 clumps of cells, each of which gives rise to an embryo and then fetus. Fraternal twins are also called dizygotic twins, abbreviated DZ, and they arise from 2 separate fertilized eggs, when 2 separate sperm cells fertilize 2 separate oocytes. Thus, they have the genetic similarity of regular siblings. So what is the genetic similarity of siblings or of parent and child? You will often hear the expression that a parent and child shares 50% of their DNA, or that siblings share 50% of their DNA. What does that mean? If we compare the raw DNA sequence of parent and child, in other words, compare their respective books of life, we will find that the DNA sequence similarity is much greater than 99.9% identical. So what does it mean to share 50% of your DNA? Here is a website that gives a nice playing card analogy to explain this concept. You should stop this video and spend 5 minutes reading this explanation at this URL. Okay, welcome back. I would like to make some modifications to this analogy. Instead of 26 playing cards, there are 20,000 different cards. Why 20,000? Because there are 20,000 genes in the human genome. Each card comes in 10 types, which represent different alleles for that card/gene. Most human genes have many more than 2 alleles, and so 10 represents a crude order of magnitude estimate. Every person has 2 sets of 20,000 cards, or 40,000 cards in total, representing the fact we are diploid and have 2 copies of every gene. These cards are placed in 2 stacks of 20,000 cards each. And then mom takes the top card from each of her two stacks and randomly picks one of them and puts that into a new stack. And does that for all 20,000 cards. Dad does the same. The two new stacks totaling 40,000 cards represents the offspring. If the offspring compares her cards to her mom's cards or dad's cards, she will find that 50% of her cards are possessed by her mom or dad, which makes sense because her mom or dad gave her 50% of her cards. If she compares her cards to her brother or sister, she will also find she will share 50% of her cards with them too. This is what it means to share 50% of your DNA. You share 50% of your alleles with your parent or your sibling. For assignment 5A, I want you to answer the question, what percent of DNA is shared between cousins? Explain. This is a hard question. Feel free to Google your answer, but make sure you explain your answer. Put this answer under question 6 of your term paper. Also under question 6, answer 2 more questions. What percent of DNA is shared by monozygotic twins? And 2, what percent of DNA is shared by dizygotic twins? Now I'm going to introduce the important concept of heritability. Roughly speaking, heritability addresses the question, how much of a complex trait is genetic? In other words, how much of the trait is due to genetic factors versus environmental factors? More formally, heritability answers the question, what fraction of total phenotypic variance can be explained by genotypic variance? The mathematical definition of heritability, which is written as big H squared or little h squared, is the genotypic variance over the phenotypic variance. This concisely captures the notion of the fraction of phenotypic variance explained by your genes or genotypic variance. Heritability equals 1 when genotype fully explains or determines phenotype. Heritability is 0 when environment fully explains or determines phenotype. As an aside, technically this is referred to as broad sense heritability. There is also narrow sense heritability, which is closely related, but we will not define here. The table shows some heritability values for various complex traits. As you can see, the heritability of some traits like height can be very close to 1. So how is heritability determined? As I mentioned before, phenotypic variance is just measured from the population. But how do you estimate genotypic variance? One way is to look at monozygotic twins, identical twins. The idea is to identify a bunch of identical twins and compare their complex trait between them. You can make a plot of the phenotype of one twin on one axis, such as height of one twin, and the phenotype of the other twin, the height of the co-twin. If each pair of twins has an identical phenotype, for example, if their heights are exactly the same, then the data point will lie on this y equals x line. For most traits, there will be a scatter around this line. You can see the scatter of the data points, reflecting the fact that identical twins don't have identical traits. The degree to which the data points lie on the line versus being scattered is called the correlation coefficient and is denoted by the variable r. And in this case, it's r sub mz for monozygotic and is called the correlation coefficient. And this correlation coefficient represents how closely correlated is the phenotype in a pair of identical twins. When they are perfectly correlated, in other words, having identical phenotypes so that the data lies on this y equals x line, then the correlation coefficient equals 1. If there is no correlation and the phenotypes of the pairs of twins are very different from one another, and so the data points don't lie on the line but form this cloud, then the correlation coefficient is 0. From theory, it can be shown that this correlation coefficient, denoted by the variable r and the subscript mz or mza for monozygotic correlation coefficient, from theory it can be shown that this correlation coefficient equals the genotypic variance, v sub g, plus the shared environmental variance, denoted by this term, v sub es, over the total phenotypic variance. And note here we're denoting variance by the letter capital V instead of sigma square. There is this new term, this VES term, and roughly speaking, it is the total environmental variance minus the environmental variance of the twins. In other words, it measures how much of the environment is shared by the two twins so that they experience similar environmental factors. More concretely, if identical twins grow up in exactly the same environment, such as the same household, then this term will become close to the total phenotypic variance. On the other hand, if the identical twins are separated at birth and they grow up in completely different environments, then this term becomes 0. So, if you only look at identical twins separated at birth so that this v sub es term equals 0, then we can get rid of it and we have that the monozygotic correlation coefficient equals the genetic variance, the genotypic variance, over the total phenotypic variance. And that's our definition of heritability. However, as we will discuss on Tuesday, there are some concerns about this assumption that the shared environmental variance is indeed 0 for twins separated at birth. And you can think about why one would have these concerns. Alternatively, a second way of estimating heritability without making the above assumption is to look at both monozygotic and dizygotic twins. The top equation is just the one we went over, the correlation coefficient for monozygotic twins, as described in slide 12. The expression for the expression for the correlation coefficient for dizygotic twins is very similar. The only difference is that the genotypic variance is multiplied by 0.5. And that represents the fact that fraternal twins share 50% of their DNA with each other instead of the 100% of the DNA that monozygotic twins share. Thus we have R sub dz equals 0.5 times the genotypic variance plus the shared environmental variance over the total phenotypic variance. Now, if we subtract this term, the dizygotic correlation coefficient, from the monozygotic correlation coefficient, then we get 0.5 times the genotypic variance over the total phenotypic variance. And then we can rewrite this as 2 times this difference, and that will equal our definition of heritability. Thus, heritability equals 2 times RMZ minus RDZ. The key point is that the twins do not have to be separated at birth. They can have a shared environmental variance that is greater than 0. This expression makes intuitive sense because this, RMZ minus RDZ is like subtracting out the effect of twins having a shared environment and just focusing on genotype, because identical and fraternal twins are expected to have similar shared environmental variance terms. Thus, there are 2 types of twin studies for estimating the heritability of a complex trait. One is identical twins separated at birth. And there, the monozygotic correlation coefficient equals heritability. 2. Identical and fraternal twins who are not necessarily separated at birth, in which case heritability equals 2 times the monozygotic correlation coefficient minus the dizygotic correlation coefficient. Remember, these correlation coefficients measure how correlated are the twins' phenotypes. In summary, in this lecture we learned what it means to share a certain percentage of DNA and how much DNA close relatives share. It means what percent of your genes do you have the same alleles. We went over the concept and definition of heritability, which is the genotypic variance over the phenotypic variance, and that captures the idea of how much of a complex trait is due to one's genes. Finally, we showed how 2 different types of twin studies can be used to estimate the heritability of a trait.